Next, we will have a special lecture especially prepared for this online event. The lecturer is Professor KAMIKOUCHI of Graduate School of Science, Division of Biological Science. If you have any questions about this special lecture, please use the Zoom Q&A session function as I mentioned before, and the questions we received will be answered online using the Q&A function during this lecture. In addition, some of the questions will be introduced and will be answered in public at the end of the lecture. Regarding the questions about sessions other than this special lecture, 4 panelists will answer them directly in the Q&A session scheduled later. Now, Professor KAMIKOUCHI, could you please start the special lecture?
Okay, thank you for your introduction. So, good afternoon, everyone, and good morning and good evening. So, my name is Azusa KAMIKOUCHI from the School of Science, Nagoya University. Can you hear me? Okay? Okay. So, it's my great pleasure being with you today. I am a professor working on neuroscience. And today, I will give you a mock lecture which is about the neurobiology of love in fruit flies. Because this is a lecture, I will expect you to ask questions. As Uemura-san mentioned, please use the Q&A function during the lecture to ask questions. A graduate student of mine, Yukina, is sitting there to answer the question as much as possible. Okay? So let me start talking about the courtship. The courtship is a way to express love. Actually, not only humans do the courtship. Many animal species, such as cats, fish, or even insects, show the courtship behavior to attract a potential mating partner. This is an example of courtship behaviors of animals. The male of this bird, a great frigate bird, displays like this to attract females. We can agree it's very impressive, but actually it's not sexy for humans. How do the female of the bird, of the frigate bird, understand such display? More generally, how does the receiver side, a female bird in this case, understand the courtship signals? We tackle this question with a fruit fly as a model. The fruit fly is a very common insect, and you can find it everywhere, maybe in your kitchen, but actually they are very useful animals in the labs to study courtship behaviors. And there are several reasons for that. Firstly, the brain of the fruit fly is very small. At the bottom, you can compare the size of brains: the human brain, mouse brain, and fly brain. And you understand the fly brain is very small. Actually, the total number of brain cells, neurons, the neurons of the fruit fly is about 100,000, which is actually much smaller than that of our brain. The second reason for using fruit fly is their molecular genetic tools, with which we can perform many kinds of experiments using fruit fly. The most important reason is here, of course, they have various courtship behaviors. With their tiny brains, many species of the fruit fly show various courtship behaviors. These are 3 examples. Drosophila melanogaster on the left Drosophila elegans in the middle, and Drosophila subobscura on the right. Each species uses a different type of courtship ritual. Melanogaster male emits a song, elegans male shows a dance, and subobscura male uses a gift to attract the female. In each species, the brain understands such species-specific signals. We are exploring the neural mechanism underlying this courtship communication of flies, especially focusing on the song signals. Okay, so let me introduce now the courtship behavior of Drosophila melanogaster. There are one male and one female in a chamber. The male, the smaller one, is now chasing a female. During the chasing, he sometimes vibrates his wing to generate a unique sound like this. This sound is known as the courtship song, which has a species-specific sound pattern, and it increases the female's receptivity for mating. So these are the songs of 4 Drosophila species. The melanogaster song is shown at the top, and the other 3 are the songs of its closely related species. The song of each species is slightly different with each other, and interestingly, the fly brain can discriminate these songs. Then, how does the fly brain evaluate sound? How do they detect if the sound is a song or just some other noises, and then if it is their own species song or other species song? Okay, before starting to talk about the mechanism for that, I'd like to introduce my research group at Nagoya University. As you know, our university is located here, and my group belongs to the School of Science at Nagoya University. This is the website of the School of Science, so please visit this if you like. I especially recommend you to watch this short movie with which you can understand more about the School of Science here at Nagoya University. Today, I will show you another video which is about our daily life in my lab. Let me now introduce again Yukina, a graduate student of mine who is a member of my lab. She is answering your question in the Q&A function, and now she will be introducing you our lab for the next 5 minutes. So please, Yukina.
Hello, guys. I am Yukina. I'm actually Japanese, but I was raised in Singapore. So anyone joining from Singapore today? Maybe from surrounding countries, Thailand, Malaysia, Indonesia? Welcome. Today, I will be your tour guide. All right, so let me start the video. So our lab is located in one of these buildings. These are the science buildings at Nagoya University. Here we arrive at Professor Azusa Kamikouchi's lab, and I work with her as a graduate student since I was in undergrad, so it's going to be my 3rd year working with her. And here she is. In our lab, we work on understanding the underlying mechanism of how the external stimuli are processed in the brain and how this shapes behaviors. And to do that, we use the fruit flies. Fruit flies are one of the most popular model organisms in diverse types of research. But why do we use such tiny fruit flies for researches on the brain? Now let's compare the brain sizes of the fruit fly, the mouse, and the human. In you may think that the brain of the fruit fly and the human looks so different. But guess what? Actually, their brains are quite similar. In fact, fruit flies also show learning behaviors just like us humans. So you might not have expected it, but a tiny fruit fly is a great model organism to study how the brain works. Using the fruit fly, we focus on the sound stimuli. On the left, males are extending their wings, vibrating them to sing a courtship song, while on the right side shows an audio recording of a courtship song. So you see a male chasing after a female continuously. Female rejects the male, but the male continues to court, never giving up. And this is very important. So we see the organism behaving like this, but we want to know what is happening in the brain. That is our question. Right, now let's sneak peek inside our lab. First, this is the student room. I work here at my desk, and sometimes I have discussions with my lab members. Now, this room is for the fruit flies. We keep hundreds and thousands of fruit flies with different genotypes. All right, and she's my lab member. She's flipping the container to provide flies with new food. And the flies are now happy, well fed. We don't only work with animals, but we use different types of machines, just like this one. We use this machine to make gene-edited flies. Here he is. He is one of my lab members, and he's looking very professional. All right, and now that is me. To select fly we want to use for our experiment, I first anesthetize the flies with CO2 gas, and under the microscope, using a smooth painting brush, I look for flies with specific phenotypes, like its eye color. Is it red? Is it white? How many hairs does it have? Are the wings curled or is it straight? Now, this required actually quite a lot of training, but once you get used to it and you get a hang of it, it's quite easy. All right, here's another experiment to see what is happening in the brain when a fly listens to a courtship song. Here we can measure the neural activity of flies when they hear a courtship song. This is the experimental setup. So we see the speaker that is playing the courtship song, and we also have a microphone to observe what is actually happening in the brain live. The activated neuron is the part that lights up like that. And this activated neuron is therefore likely to be important for processing the courtship song. If so, what happens if we gene edit the neuron that lit up in the previous video to lose its function? To answer this, we use another set of machines. By performing different types of experiments, one at a time, we hope to reveal an unanswered question of how our brains work. Fascinating, isn't it? If you got excited after listening to our lab tour, come join us at Nagoya University and join our family. We're waiting here with open arms to welcome you all, and here are some bloopers. All right, that is the end of the lab tour, and I'll pass back to my professor.
Thank you, Yukina, for helping me to introduce my lab. Actually, she talked a lot, almost everything. But anyway, I will start my lecture again. Okay, so I go back to the question. How does the fly brain evaluate sound? This is a flow of sound information processing in general. We process sound signals with these three systems: the filter system, decoding system, and command system. The ear and the brain are involved in these systems, and auditory sensory cells, auditory interneurons, and command neurons are responsible for them. We are studying filter ear and the brain to understand the organization of these systems. But you may wonder how the fruit fly hears sound. This is a comparison between human ear and the fly ear. We have an eardrum at the ear located here as a sound receiver. In the cochlea, there are auditory sensory cells to detect the sound stimuli received at the eardrum. This is the human ear. On the other hand, the fly ear is located at the antenna. The arista sticking out of the distal part of the antenna receives sound stimuli. The basal part of the antenna houses an auditory organ called Johnston's organ, and there are auditory sensory cells in it. Then, how can we explore the organization of the fly ear? The answer lies in the genetics. The genetics is a tool we can use for experiments. The GALPHO-UA system is one of such tools and probably the most famous and useful tools. Here, just by crossing two types of flies, we can express any genes in any cells of interest. For example, if you want to express this red protein in this neuron, you can do so by a single fly cross, and then you will get flies in which the neurons is visualized with red protein. With such molecular genetic tools, we can analyze the anatomy of a specific neuron by expressing some reporter proteins such as green fluorescent protein, GFP, or DS-red, red fluorescent proteins. We can also visualize the neuron's response pattern by expressing these indicators, GCAMP or chameleon, each of which indicates the neural activity. Contribution of each neuron to a specific behavior, mating behavior in this case, can be tested by expressing effector proteins that can manipulate the activity of the neurons. This is how the Johnston's organ, the fly ear, looks like. Using genetics, we expressed GFP, a green fluorescent protein, to visualize sensory neurons in the Johnston's organ. You can see that each of the round-shaped cell body of the sensory neurons, labeled in green, send a thin fiber from left to right in this image. This fiber is called an axon, which transmits signals to the brain. This bottom image shows the fly brain, the frontal view of the fly brain, in which the green labeled axons of sensory neurons coming from the fly ear are visualized. The sound information detected at the fly ear is transmitted to this ventral side of the brain. So, what would you do if you want to explore this question? How does the fly understand sound? We used these fluorescent proteins to visualize the neural pathway from the fly ear to the brain and also neural circuits in the brain. What we did was a large-scale screening of neurons that fibers that send neuron fibers in the primary auditory center in the fly brain. We used a lot of gear-4 fly strains to perform a mosaic analysis, a kind of agentic analysis, to visualize different sets of neurons in the brain. And this is one of my favorite neurons in the brain. This is the round-shaped one is a cell body, which has some gorgeous fibers in the visual center, primary auditory center, and a brain region known as GNG. And from its morphology, we can predict that this neuron sends signals from these two centers, visual center and primary auditory center, to the GNG. After identifying neurons, as Yukina explained, we can monitor their responses to sound stimuli. Calcium imaging experiments allows us to monitor the neural activity to a given stimulus in alive animals. We express a special protein, GCAMP, that shows a fluorescent increase when the neurons are activated, and then start sound playback. A fly that expresses GCAMP protein in auditory neurons, sound-responsive neurons, are placed under the microscope like this. We made artificial fly songs and other sounds for the playback and tested the fly. If these neurons respond to the sound, you will see this fluorescent increase here in the brain, and then get to know what sound type activates these neurons. Okay, we have now looked at the neuroanatomy and response pattern of the neurons. What's next? The final piece here is the behavior, to ask how the identified neurons contribute to a specific type of behaviors. To evaluate the relationship between the neurons and the behavior, a good strategy is to manipulate the neurons and monitor the behavior. This is one of the tools to manipulate neurons we use. Tetanus toxin is a neurotoxin that inhibits neuronal transmission. When we express this toxin in this neuron loses its ability to transmit signals to the next downstream neuron. What would happen if we do so and in a behaving fly? To monitor the behavior, there are several assays to check the fly's behavioral response to sound. The left side movie is a song-induced chasing test for testing males, and the right is a song-induced receptivity test for testing females. In either test, when the flies detect a favorite song, their behavioral response increases. You can see that in the left, the male test, the flies are chasing each other. And in the right, two flies are working together in each chamber. This is the experimental setup for the assays of males and females. We put flies in a chamber and play back a sound file from a loudspeaker here or here. For the male assay, we put 6 males in each chamber. When these males detect a favorite song, they start chasing each other. For the female assay, there are 1 female and 1 male in each chamber. The male is chasing a female and shows courtship behavior. But because we artificially removed his wing before the experiment, he cannot sing a song for her. And courtship always fails. But to help his courtship, we play a song from a loudspeaker. When females detect a favorite song, she gradually started accepting the male's courtship. We can use artificial courtship songs here for the playback. Video camera here records their behaviors, and we can analyze later the movie file to see if the flies show the behavioral response to sound. Using all of these tools and assays, now we have understood the early stage of auditory information processing in fruit flies. The fly ear has 3 types of auditory sensory neurons, by which frequency information is segregated. One type, JOA neurons, detect high-frequency sound. The second type, JOB neurons, are tuned to low-frequency sound. And the other one, JOD neurons, are detecting middle-range frequency sound. Overall, the frequency information of sound is segregated at the level of the fly ear, which is quite similar with our human ear. In the brain, there are many secondary neurons so far identified, at least 44 types or more. And the frequency information are integrated at this level. And the left side and right side information detected at the left and right ears, as well as other sensory information, such as olfactory, smell, and taste, and vision, are integrated at this level. Some of these neurons send information to the song processing pathway, and the other for the escape pathway. And these are our recent findings here at Nagoya University. The first one is how they detect the song rhythm, and there is a special type of neural circuit contributes to it. The second one is a finding that fly learn the song rhythm, which means that they have a plasticity in their song processing pathway in the brain. The third one is a neural mechanism of the female's behavioral change from rejection to acceptance in their mating behaviors. We are getting more and more findings now, and I'm happy to share them with you in the near future. Okay, from now on, I will talk a little bit more about this finding. The flies learn the song rhythm. Some minutes ago in this lecture, I talked about the courtship song of flies, and then they can tell the difference between the song of flies species. The question is, what parameters they are evaluating in the song? So these are the songs again, and I will explain it now in more detail. The major difference between these songs lies in the song part known as the pulse song here. The interval between pulses, interpulse interval, is different between these species. The melanogaster song shown on top has a faster rhythm than other 3 species songs, and they can discriminate a rhythm defined as the interpulse interval, IPI, which is about 35 milliseconds in melanogaster, and longer in other 3 sister species. Dr. Lee, who got a PhD some years ago in my lab, had come up with a brilliant idea about the question, how the fly brain discriminates the IPI, the song rhythm. His hypothesis was that experience of hearing a good song may be the key event to tune the IPI preference in flies. To test this hypothesis, we set up an experiment of giving the flies the experience of hearing a good song in the lab. We made 2 types of artificial songs using computer. The first one is a good song, which mimics the same species song, carrying a 35 millisecond IPI. The other is a bad song, which has much slower rhythm, mimicking other species song. This is how we did the experiment. We collected the flies directly after hatching and subjected these young flies to a training in which flies can experience a good song or a bad song. We put single flies in a small chamber like this and placed it in front of a loudspeaker. Actually, we divided the flies into 3 groups shown here for the training session. For the first group, shown in blue, we do not play any songs during the training session, which lasts 6 days. So these blue group flies have no experiences of hearing any songs. For the second group, shown in red, we played a good song during the training. A good song means the same species song. For this orange group, we played a bad song mimicking other species song. We gave them such experiences, which we call training, for 6 days, and then tested their response to each song. Again, we used these tests to see the song preference of flies in each group. For both males and females, when they detect a favorite or a good song for them, their responses would go up. This is the result. So we have 3 groups at the training session: blue, red, and orange one. So we use this fly to the song preference test. Here, the x-axis of these graphs here represents the amount of flies' response to the song we gave at the test, the same species song or other species song. And the color of each bar corresponds to the group at the training. At the song preference test, the response to the same species song shown here on the left are not dramatically different between these 3 groups. On the other hand, the response to the other species song showed a dramatic decrease in the red group, which was trained with the same species song. So this means that experience of hearing a good song makes the flies more choosy, and now they can choose only the good song. So we also found that training with a good song, the same species song, good training requires GABA, an inhibitory neurotransmitter widely used in both humans and insect brains. We found this GABA recruitment in the brain suppresses the fly's response to other species song. Okay, this is a summary of this finding. We have found a phenomenon that experiences of hearing a good song can tune the song preference in flies. In other words, with proper auditory experiences, they can refuse the song of another species. We also identified the neural mechanism for this behavior plasticity, which is shown here. There are some special neural circuits which involve GABA work for the experience-dependent behavioral changes. We now expect that the phenomenon and its underlying neural mechanism may share some similarities with the language learning in humans and song learning in songbirds. Okay, it's almost the end of my lecture. At the beginning, I talked about the courtship, which is a way to express love. At the receiver side, you now realized that the brain is working hard to evaluate the courtship signals and decide to accept love or not. Finally, I'd like to acknowledge these people who contributed to this mock lecture. Thank you so much for your kind attention, and I'm happy to take questions from you. Okay, so I got some questions you posted at Q&A function. The first question is, how long have you conducted research? So about myself, okay, so I've started my research. It was 20 years ago. So first I started with the honeybee brain. I was studying honeybee brain about their communication language. So honeybee have a dance language to communicate with each other, and I was very interested in the honeybee brain, how their brain can do that. And then I started to use fruit fly to understand the fruit fly communication system. And I've been working on it, on the insect brain, for 20 years so far. Okay, the next question is, how long does this experiment take to make a conclusion? Which experiment do you think?
Maybe female receptivity.
Female receptivity test. So Dr. Lee's experiment took him 3 years. So from the hypothesis and the conclusion and publish a paper, it takes 3 years. And I think, in my opinion, so 3 years is the average time to do one project for us, at least in our project. Okay? So the last question, how the male fly can recognize the specific female fly? You mean the independent fly in the same species? Okay, so the answer is I don't know. But I'm not sure the male fly recognizes individual females. So it looks like he's just chasing the female close to him. So there are not so many females in the field. So if he finds a female, he has to chase the female. That is my impression, but I'm not sure. We have to ask the fruit fly for the answer. Okay, it's almost time. I'm going to close my talk. Thank you.